and explain this to other people because it's so critically important that we stop killing them and we start putting them back into our soils. Those of you who like to go back to the original science, this is the paper, the original paper about nutrient cycling with the organisms in the soil. This is the most referenced paper in the world of soil ecology, microbial ecology, because here's where we first established bacteria and fungi being fed by the exudates coming out of plants, solubilizing nutrients out of the sand, your silts, your clays, your rocks, your pebbles, tying them up in the bacteria and fungi. And the protozoa, nematodes, microarthropods, eating them, releasing the nutrients, plants grow better. I do like to point out to you that this is a really good paper. It's dense science. And so maybe you'd rather listen to me because I try to make this way more understandable by the average public. But those scientists in this audience, this is the original paper. Now I do like to point out here that I am not this Ingham. That's my husband. I'm the third author on this paper. So you can imagine, my husband is a nematologist, so he did the work on the nematodes. I did the work on the bacteria and fungi and protozoa, along with a herd of other people. We had about 12 scientists working on this project, because when we started this project, nobody really understood why protozoa were important. So what? You got protozoa in your soil. What does it matter? Nematodes. They have to be there for a reason, right? Or Mother Nature wouldn't keep them there. But we didn't know. So here's the data. Here's all the dense science, and it's a great way to put yourself to sleep at night. If we're suppressing the diseases, by having those organisms that prevent the disease-causing organisms from being able to grow. If we have our food web, we build soil structure and we get oxygen moving all the way down in soil. How far down does soil go? 16 miles.
To China.
Well, it doesn't go all the way to China. We have that little problem called the center of the planet is molten. If you took the right angle, maybe. So how far down have we found active living bacteria, fungi, protozoa, nematodes, microarthropods in soil? So working with some of the oil companies, when they're drilling deep and they're bringing back these cores, and we start looking through our microscopes, the same ones that you can buy, we see these living organisms present all the way down to 16 miles. Now, are your roots going to go 16 miles? We haven't really been looking for that. But typically not. How far down do roots go? So hold on, we'll get there. Y'all have to stay awake. So if we're getting these really good aerobic organisms growing, protecting our root systems, there is no need for pesticides. You would not ever make an anaerobic tea in order to make something so toxic. It would kill the bad guys, because you're going to kill the good guys too. So anaerobic, really stupid idea. Now, how about your digestive system? Is it good for you to eat fermented foods? Yeah, because guess what? Your digestive system is anaerobic. So when you poop and manure comes out, what are the organisms growing in your manure? Anaerobic. And so we gotta compost that material. So cow manure, chicken manure, any kind of manure is coming out of an anaerobic environment, and now we have to convert those organisms to something beneficial to your soil that will do these jobs. And it doesn't take much, but you do have to do that conversion. Applying manure onto your soil is not a good idea unless you've got a really healthy set of microorganisms. And even then, I would want you to mix the manure with something else to get as much oxygen in there before you go out and put it and compost in place. So, retain nutrients. Hopefully now you all understand exactly why that's happening when we get the really good biology. And of course, if we're retaining, we're not losing any of our soluble nutrients into your drinking water. The lakes and rivers and streams, that whole problem has gone away. When we're looking at making those nutrients available, now you understand that the predators in the system are required, so that right there around the root system, nutrients are going to be made available to your to your plants. What happens if more available nutrients are made for your plants than the plant can actually take up? What happens to those soluble nutrients? Well, who's right there? All the bacteria and fungi, and they just go, well, the plant didn't take it up, so I get it. And so we're holding those nutrients right there. If you've got some compaction problems, if you've got anaerobic conditions happening, by getting this good biology back in there, we're going to wipe out, they're going to use all those toxic materials. So building soil structure. If we're building structure in our soil and we're holding our water in these little pools of water throughout that good structure, we reduce, typically in the very first year, we reduce water use by 70% as compared to conventional systems. And haven't you in the permaculture world seen some of that already? Yeah, but let's get there within 3 months. Let's get there in 21 days. So we hurry up and speed up that process so you don't have a 3-year transition time, so you don't have a 5-year transition time. If you understand your life and your soil, we get you there really fast. If you're kind of trialing and erroring it, can take a while because you're crossing your fingers and hoping every time you apply something. We want our roots to go down as deep as they possibly can go, and that's usually a big surprise to people. How far down can roots of the grasses in your lawn, how far down can they go? Well, stay awake, we'll get there. So, just a couple pictures of these organisms, and these are using the $350 microscopes. If you don't want the camera, they're only about $280, so kind of up to you. And generally, if you go to the company, I'm gonna zip, there you are. That's the company that you would order this from, microscopenet.com. And when you contact them, tell them that Elaine Ingham told you all about that website, and they will typically give you a 10% discount. So, have some fun with this one. $350 microscope plus camera, and the last time I went on the website, like 2 days ago, they had a special on. They were selling these microscopes for $280 with the cameras. So, go and find them. These have LED lamps in them, so that you don't have to buy additional lamps. These lamps will last your whole lifetime. So, really good deal. So, going back, can I go backwards? There we go. So, looking at this nematode right here. The way we're going to identify that this is a beneficial nematode is based on how it looks. Turn around and look at the organism next to you. How do you know that's a human being? How do you know? They look like a human being, right? So we identify organisms on the basis of morphology. So head, arms, legs, okay, I think you'll agree with me that this is a human being. We think. What kind of human being? Is he a good guy or is he a bad guy? Well, look at the habitat that he's in. He's a good guy. Has to be a good guy. Good aerobic environment, freedom to think and talk and kick ideas around. Good guy organism. So we're going to look at this nematode, and that mouth, there are no spears. Simple mouth. All this guy is doing is sucking in the soil solution, eating bacteria. So nice, simple mouth, no teeth, no spears, no knobs. It's bacterial feeder. This is Elamus. Isn't that a great name? She eats bacteria. And whenever she eats bacteria, there's too much nutrient in those bacteria that she's eating. So she's releasing this balanced set of plant available nutrients for your plant. So how did we prepare this slide? We have cute little test tubes that you buy. They're not very expensive. They're like $0.10 a piece. And if you get 4 or 5 of these little test tubes, they're going to last you for the next 50 years. So not a big expense. We're going to put a teaspoon of our soil or our compost or whatever solid we want to look at. Add 4 teaspoons of water. Shake it for about 30 seconds gently. break up all of the water-stable aggregates. We're then going to take an eyedropper and take a drop out of that 1:5 dilution. Put that drop on our microscope slide, cover it with a cover slip that goes on the stage of my microscope. Turn the light on my microscope and I look through there and I go, whoa, good deal, I've got good soil. It's about that simple. Now, do you have one of them in every field? Or, gosh, it takes you 100 fields to find another one. So, if it takes you 100 fields to find another good organism, should you be feeling really good about your soil or kind of sad? Sad. If you're finding one of these in every field, whoa, you should start selling that stuff. So really good stuff. How do we figure this out? It doesn't take very long. It takes you 15 minutes to try to actually determine, do you have the organisms? So when we're looking at this field of view, who are these critters in here? So we've got a micro aggregate here. You can see some rod-shaped bacteria. They're longer, they're rods. We got some little cockeyed, round guys. You didn't know you were coming here to learn Latin, did you? Cockeyed, Latin for round. Bacillus, Latin for rod-shaped. So we've got a nice little aggregate that's been built here. Well, there's another one, and another, and another. These have, in the past, been bound together by some good guy fungi, but look at the fact that there's no fungi in this picture. We've lost our fungi. And so as a result, this tells you that in the past, I've had a good soil, I've had good structure, but I have lost a critical component. I don't have that fungal biomass, those filaments of fungi in my soil. And as a result, when I'm looking at my root here, can you see this divot in the side of the root? See where a bite has been taken out? This is actually a fungal disease attacking that root system. And we know in approximately 4 days, this will be a dead plant. So get advance notice that the bad guy fungi are taking over. And the bad guy fungi are not filamentous. Bad guy fungi are the yeasts. The anaerobic fungi, the anaerobic bacteria are attacking and consuming your root system. You've got time to make a compost tea and get those organisms to rebuild the structure and get the oxygen back in here and put those bad guy organisms to sleep. But you gotta be monitoring your soil so you know that bad things are starting to happen. And when we look at this organism right here, this is a protozoan. It eats bacteria. See all those cute little bacteria in its digestive system? Want to count those bacteria? Millions. This is a ciliate. It's the protozoan that grows when oxygen starts to drop to a low level. So nature is sending you a message. Uh-oh, things are going bad. So we have three things in this picture. They tell us, it's time to start working and getting this life back into your soil. Only starts to outcompete everything else under reduced oxygen conditions. And we identify him based on it's a single cell. And see all those cute little cilia? All those little hairs? So we're going to be, you working on our microscope, focusing, pushing the iris diaphragm, shadowing, so we can see those little hairs, and then we can start crying. Because we got work to do to get it back out there and rebuild the beneficial life in this sample. So keep going. Fungal hyphae. And so when we start looking at fungal hyphae, notice that some of these hyphae are out of focus. Because when we put that drop on our microscope slide, put the cover slip on top of it, we've got 15 micrometers depth that we've got to look through. And these organisms are on the order of, well, if we're looking at bacteria, that's about a micrometer in size. The smallest, littlest round cocci in here are a micrometer in size. So if we're trying to figure out what's the diameter of that fungal hyphae, we go to the place where it's actually nice and in focus. The edges are real sharp. Now, how many of those little bacteria could we line up across there? Yeah, and try to hold your hand steady here. 1, 2, So, take a look at that guy right there. So, 1, 2, 3, 4. What's the diameter of that fungal hypha? 4 micrometers. So is that good or is that bad? Any fungus, any filamentous fungus with a diameter over 3 is most likely going to be a beneficial fungus. The bad guys are skinny. They're narrow. They're only 2 micrometers typically. So when we're looking at fungal hypha, you can see it's uniform diameter all the way along. The branch may be slightly different. That's okay. But this is uniform all the way along. Now, people often look at this guy and say it's not a fungus. Yes, it is. Because this is a lot closer to your eyeballs. The part that's in focus right there, that's where it's in focus, and then this is out of focus. So you got to use the focus, and you got to run along that organism and determine that, yes, it is uniform diameter. Yes, it is uniform diameter. Now, there are actually 4 species of fungi in this picture. Can you find them? Because I pointed out this guy, beneficial, nice, uniform diameter all the way along, wide diameter, it's greater than 3. Oh yeah, and it's dark in color. Colored fungi are the best. Anything colored is most likely a beneficial fungus. So, very beneficial. See all that little grainy stuff on the surface? It's instantaneous ability to hold soluble nutrients. Binds on the surface. This is like the pantry for these fungi. They're going to keep all the nutrients until the plant says, hey, fungus, I need some whatever. And then the fungus takes its enzymes and just pulls it off the oxalate crystals that it produces on the surface of the hyphae. How many of you found that fungal hypha right there? Oh, it's clear. It's colorless. So probably not as good as this guy. But what's the diameter here? Yeah, it's wider than this one. So, how would you like to estimate? About a 6, would you say? So 6 micrometers diameter, clear, colorless. So where does it rank on goodness, badness? Yeah, it's still good. Now, probably not quite as beneficial to your plant as this guy, but it's still on the good guys. It's not a disease. How many of you found this guy over here in the corner? Oh, tricky. See him right here? Sorry, way in the back, but it's hard to see. But what's diameter? About a 3? Is it colored? Very lightly colored. It's a tan. But because it's tan, it's darker in color than the background, and what you always want to compare to is the background. It's probably a beneficial fungus, maybe not quite so good. Now, where's the bad guy fungus in here? He's a little out of focus. It's hard to focus on everything in here. But you see this really narrow diameter, clear, colorless, about 2 micrometers in diameter. That's Pythium. In this particular instance, we took this sample and plated it on a plate medium, and the foods in the medium in that dish strictly grow Pythium. And we got a boatload of Pythium growing from this soil. So we knew the bad guy fungus was Pythium. That's what grew. But should we be worrying about that? You see, you send this soil into a plate count lab, and they're going to go, oh my God, you've got a horrible disease problem. You better get out there and nuke it with a fungicide because it's just going to take over your plant and you're not going to have any production at all. And so you, oh gosh, and you go buy the fungicide and get out there. In the organic world, what's our fungicide? Compost. Copper sulfate. In the chemical world, compost is the way better. The compost is the right answer there. But you see, if we use copper sulfate in the soil, we would kill all of the beneficials. And we would not kill this Pythium because it's now resistant to both copper and sulfur. So, okay. Now, do we really need to be concerned about it? No. Because if we've got all these beneficial fungi, they are going to prevent the growth. We're going to keep the structure in the soil, so oxygen's going to move into that soil, and that Pythium's going to go, my enzymes don't work when the oxygen concentration is this high. So I don't have to worry. If you've got the beneficial organisms there, they do the work to make sure the habitat is beneficial. The good guy organisms will grow. So, why be concerned with soil life? I hope you can start figuring that one out. Don't, We're gonna get the proper balance of nutrients so the plants taste good, they satisfy your hunger. You only have to take one bite of kelp and your body's going, okay, I got all the nutrients I need, and it satisfies your hunger. If your body's not getting the nutrients that it needs, it keeps saying, feed me, feed me. I don't have the iron, I don't have the zinc, I don't have the whatever it needs. And so you keep feeling hungry, and so you eat more empty calories and more and more. Not very beneficial for us or for our animals. So need to have the nutrients in the plant. Where do plants get their nutrients? Well, it's from the biology, and I promised that I would show you nutrients present, the mineral present in soils. And when you look at soils all over the world, there is no soil, there is no dirt on this planet that lacks the nutrients to grow plants. And the data are there in any introductory soil science textbook you want to look at. The data are there, and we've known this for the last 150 years. And yet soil scientists disregard this. You've got all the phosphorus you could possibly use. If you could take the phosphorus from a single grain of sand, you would have enough phosphorus to grow every plant in this acre of agricultural soil. Well, you know, but how do you disperse all the phosphorus in a grain of sand out to all of the plants in an acre? So that's why we have sand throughout all of this. And we're taking a little bit from every grain of sand. And it is the bacteria and fungi that make the enzymes to pull those nutrients out of your sand, your silt, your clay, your rocks, your pebbles, your bedrock, organic matter. You will not need to put any fertilizers out as long as you have sand, silt, clay, rocks, pebbles, organic matter. So any of you run out of rocks in your soils yet? So no need to put out inorganic minerals. Not required. If your plants are showing a lack of nutrition, it's because you lack life. Test your soil, figure out what's missing so you can get this normal natural nutrient cycling process going. Now, when we look in the soil and the Earth's crust, when we look in sediments, there is no nutrient that's lacking. Now, some of them look a little bit low, but that's molybdenum. Your plant needs nanograms, not micrograms. So we've got hundreds of years of every mineral present in our soils. And we got to exit that idea that, oh, you got poor soil. You don't have enough phosphorus. Excuse me, you do too. What you lack is the biology to convert it into phosphate available for your plant. It's fix the biology, not go out and buy something you don't need. You don't need to be buying those rock dusts. You don't need to be buying lime. You don't need gypsum. Why are you buying gypsum? Why are you buying lime and putting that out on your soil? Well, my soil's pH is down at 4.5. What's causing that pH to be at 4.5? And if you put lime on it, what's going to happen in about 4 weeks? If you put lime on an anaerobic environment, have you fixed the anaerobic condition in that soil? No. No, you put some material on there that will neutralize the acid, but then what's the biology doing? It's going to make more acid. So in 4 weeks, your pH is going to be right back down to 4.5. Ooh, your plant got a month worth of normal growth. Well, is it significant? Sure it is. But shouldn't your plant be getting all the nutrients that it needs during the whole growing season and not just for a month? So, got to get the life back into that soil. So, life, we've got to get that back out there. I do like to point this out because when you send a sample into a soil chemistry lab, you send your sample into a lab to get the nutrients done. What are you actually being told about? When you look at, there's the total amount of phosphorus in your soil. It is the bacteria, fungi, protozoa, nematodes, microarthropods that convert that into the ionic form, phosphate. And some of that's going to tie up on the surfaces of your sandstones, clays, rocks, pebbles, organic matter. It is the biology that takes that surface-bound material and converts it into soluble forms, PO4, in your soil present in solution that your plant can take up. But your soil chemistry report only tells you about this. That teeny, tiny, little bit of soluble, available phosphate. That's all they're telling you about. And can I scare you? If all I'm telling you about is this little bit of that nutrient, can I scare you? Oh my gosh, you don't have enough phosphate in your soil. You better buy my inorganic fertilizer because otherwise you're not going to be able to make a crop. Scare you to death. And we bought into it for the last 70 years. And we've gone out and put this stuff out there. I love it when somebody says, we're running out of soluble phosphate deposits on the planet. What are we going to do in 10 years when the inorganic phosphate supplies run out?
Cheer.
Yeah, cheer. Yeah. Because then we're going to be forced to go back to the organic way of doing things. So realize that in your soil chemistry report. They're only telling you about that. When really, this is what you should know. Or really, why are we doing soil chemistry tests? Because I can show you soils where there is absolutely no measurable phosphate. My phosphate levels in this old growth forest are down at 0.00001 part per million. And any agronomist will go ballistic if you say, I want to grow plants in here. And yet when you look at the tissue of that plant, it is at high levels of phosphate in the tissue. It's not the concentration of nutrient that's present right now. Because who's cycling and making more nutrition, more phosphate available to that plant in the next second?
Bacteria, fungi, protozoa, nematodes,
Very
microbes.
good. Excellent. Need to hire her. How many extracting agents are there for this pool of nutrients? How many for that pool of nutrients? How many for that pool of nutrients? So typically, there are about 1,000 different ways to extract soluble. Which extracting agents are actually giving you truth? What your plant's actually going to take up? And only the plant knows for sure. You know, if we're using stronger extracting agents, we're finding about this pool. How many different extracting agents give you the exchangeable pool information? There's 650. Now, this is not me coming up with these numbers. I talk to soil chemists who run soil chemistry laboratories, and this is the information they give me. 650 different extracting agents that give you this information. Which one's true? Which one's right? Which one's telling you? God only knows. And God's not talking. So if we take really strong acids, if we take 24 molar hydrochloric, or we take 12 molar sulfuric acid, grind your soil up really, really fine to micronized particles, and we extract with these really strong acids, and we run that through GC or HPLC or mass specs, whatever you want, we will get this information. But how many soil chemistry reports, if you send it into a laboratory and ask for nutrients, how many laboratories give you this information? None of them tell you that information. You have to ask for that special. You've got to ask for the total nutrients in your soil. And guess what? It's already been done. Go to the USDA NRCS soil database, and they have already looked at what your total levels are. So why do you have to pay that money and do it again? Have the total nutrients in your bedrock, your sands, your silts, your clays, your rocks, your pebbles, has that changed? No. Stop wasting your money. Go out and do your biology. Learn to use a microscope. And you can do that yourself. You can figure out what your biology is yourself. It would be great if you would all send the samples into the Soil Food Web laboratories, in New York and Oregon and South Africa and whatever. But it takes 2 weeks for you to get the information back. How useful is that for you? 2 weeks. Has the biology in your soil changed in 2 weeks?
Yeah.
Of course it has. And who changed it?
Plants.
Exactly. The plants did it. Now here we're looking at how much of this is actually the root system of that pine seedling, and how much is the life that you can see. So here's our pine seedling. There's one root going down. There's another, and there's another. All the rest of this is mycorrhizal hyphae, mycorrhizal fungi. Look at how much your root has actually expanded when it gets colonized with life. Building structure, holding water, holding nutrients. And with the mycorrhizal fungus, this is an ectomycorrhizal fungus. It's actually going out and solubilizing and bringing directly back into that root system. All of our crops, all of the plants we really care about, the things that grow food for us, except for the brassicas, except for the cole and kale crops, everything else requires mycorrhizal fungi to be healthy, to be happy, to give you the nutrition that you require for your animals and for yourself. So we want these mycorrhizal fungi. What if you're growing brassicas? What if you're, well, those crops require the microorganisms that prevent mycorrhizal fungi from being able to colonize them. And so when we're looking at those plants, what microorganisms prevent mycorrhizal colonization of the root system? They're called actinobacteria. So do we need actinobacteria in our compost? Or do we need the mycorrhizal fungi? Okay, that's our compost. Invite me back for a compost day and we can go through that.